Welcome to Recovering Evangelicals, a podcast for people who were once very comfortable in the Christian faith until the 21st century intruded and made it very hard to keep on believing. I'm your host, Luke Jansen.
And I'm Scott Dyer.
Today we're going to take a journey through time looking at the transformation of four-legged reptiles having molars and incisors into slithering, legless snakes with syringe fangs that inject a complex soup of toxins into their prey. Not necessarily to kill the prey, but to at least immobilize it so it can even swallow the prey live. This was not a rapid change, nor was it a simple change. It took a complete transformation of the body over millions of years, as told by the fossil evidence. Beginning with the loss of limbs, the addition of many more vertebrae and ribs, then the transformation of a salivary gland into a potent producer of venom, the reshaping of mere incisor teeth, like your dog's teeth, into long, curved, and hollow fangs, much like a syringe needle. And finally, we'll look at a more recent adaptive behavioral response to deal with pesky primates and hominids who can't resist the urge to beat the animals with sticks. And all of these anatomical and biochemical changes require more than just one or two spontaneous mutations in the genetic code. So we're going to take a glimpse at the complex coding behind this transformation as well. But to do all this would require a truly well-informed guest, because what do Scott and I know? Well, we hit the bullseye here. Dr. Bryan Fry is a professor of toxicology at the University of Queensland in Brisbane, Australia, a world-renowned venomologist who studies the evolution and biochemistry of venoms from snakes, spiders, and other creatures. In addition to his many scientific papers and 2 popular books, he's featured in over 100 documentaries on platforms that include Animal Planet, the Discovery Channel, National Geographic, and the BBC. And now he can add to that list the fact that he's also featured on our own podcast. Bryan, welcome to our podcast.
Thank you for having me.
I got to tell you, Bryan, just in doing some background on you and so on to familiarize myself, I really enjoy the many documentaries and videos, just absolutely fascinating. I've had a long-standing interest in reptiles. Not so much snakes, but certainly that as well, and marine life. I was the founder of the Marine Aquarium Society of Toronto many years ago, and so it's always been a dream of mine to go to Australia, which, the mecca of all places for marine life. But I got to say that it was really hard watching your documentary because you're handling these snakes, and you're talking about how one bite can just absolutely devastate you. And yet, you're handling them, you're looking them right in the eye. Like, I'm thinking, like, how do you do that? How do you know that snake is not just going to get pissed off and lash out?
Well, like everything, it's a calculated risk, and when it comes to handling the snakes, we have a simple philosophy that casualness leads to casualties. So by that token, by the time that you start becoming complacent, that's when things get really, really dangerous. And you should basically stop working with the snakes. So you should always be, a little bit alert, but not alarmed, you might say.
Okay.
And what got you so interested in a career that involves crawling into and diving into small, dark spaces with potentially lethal animals like these things?
Well, I was a really weird kid, but I suppose my lifelong fascination with toxins was perhaps inevitable, because my very first memories are actually being in the hospital in absolute agony with spinal meningitis that I had picked up at daycare. And if you're not familiar with it, this is a disease that even today strikes cold fear in the heart of doctors and parents alike, because it causes a severe inflammation of the lining of the spinal cord and the brain, and it's highly lethal. Like, there was a outbreak here in Queensland a few years back where I think it was 14 kids at a summer camp came down with it, and 6 of them died. So in my case, I was left with permanent deafness in my right ear and pretty dodgy balance, because it's one of those diseases that if you do survive, you're very unlikely to get through it unscathed. Like Helen Keller, the famous disability advocate, who was deaf and blind, she lost those senses from having spinal meningitis as well. So it's one of those ones where, it is such a shaping experience. So in my case, I became, intrigued by this idea of these invisible substances that can just wreak havoc on your body. And when I was released from the hospital, my muscles were so wasted and damaged, I had to relearn how to walk, all over again, and spent a lot of time bedridden during that process because my poor little muscles would just get so very, very tired. And during that period, I would devour all the books I could get on snakes, and particularly venomous snakes, and became enchanted with this idea that they were able to inject you with substances that would cause all these deleterious effects on the body, some of which were not dissimilar to the spinal meningitis that had, caused so much damage to me. So there's this natural intersection between that formative experience and then, becoming just seduced by the beauty of snakes.
Bryan, I have to ask, so you did start this story with you as a child, and now I have to wonder, did Marvel Comics have anything to do with this? Because they introduced that character, Dr. Venom, to scientists who work for a group called Cobra, and when I did the math, you would have been about 10 years old when they brought him out. Was Marvel Comics your gateway into this career?
Yeah, I hope that I got superpowers out of all this, but tragically I didn't, other than maybe weapons grade OCD. Yeah, I was of course a complete geek, like most kids, and devoured all those comics as and X-Men and all that. But one of the things that really does drive me is a huge case of survivor's guilt, which is something that's totally illogical but quite common. Like you see it all the time in the military, where someone's in a Humvee and it gets blown up by an IED and they're the only one of their squad that survives. They're left with that feeling of unworthiness. Why did they survive and others didn't? In my case, why did I survive that outbreak when other little kids died during it? So one of the things that fuels me is to try to alleviate suffering in others, to help sort of get rid of a bit of that feeling of unworthiness inside of me. So that's one of the reasons that a lot of our work is very focused on the drug design and development coming out of novel toxins, but also studying how well antivenoms work and other treatments for envenomation.
Well, Bryan, you not only survived meningitis, but you also survived a snake bite. Actually, you survived, last I heard, 27 snake bites, and you tell a really great story of your first one. It's got to be an occupational hazard for you, but maybe tell us a bit about that experience.
Yeah, so the current career tally at the moment is I've milked, extracted venom from over 30,000 snakes. And of course, things do happen, particularly earlier on in a career. So yeah, the body telling the story stands at the moment of 27 snake bites, 25 broken bones, including breaking my back on 2 separate occasions in 3 places the first time, 3 concussions, over 400 stitches, about 60 of which I put in myself, and of that, about 20 without local anesthesia. That hurts. 2 stingrays, nearly being killed by a scorpion deep in the Amazon. So yeah, there's been a lot of things along the way, not all of which were avoidable, some of which were from being just very, stupid as a 20-year-old. I know that, 20-year-old males are their own worst enemies. But yeah, the very first one, like many such, first events, was very memorable. And in this one, I was growing up, we had a house full of all kinds of critters, but nothing venomous. And we had an agreement that when I turned 18, that I could get venomous snakes. And I was still living in the States at the time. So not long after my 18th birthday, drove out to a mate's farm in rural Oregon. We were living in Portland, Oregon at the time, and looked over his collection of rattlesnakes, picked out a few to buy from him, and in the process of this, managed to get bit by a really pretty sunset pink and black canebrake rattlesnake from northern Florida. This very unique population that's just spectacularly colored, just absolutely beautiful, instead of the normal tan and black that you get from the canebrake / timber rattlesnake clade. Now, of course, like many 18-year-old males, I was a know-it-all, and I had read everything I could find on rattlesnake envenomations up until then. So I knew with absolute certainty that rattlesnake bites are always characterized by rapid, intense local pain and swelling. So with the risk assessment capacity for which 18-year-old boys the world over are famous for, we didn't seek immediate medical attention. Instead, we just sat in his parents' kitchen, who weren't home at the time, and just waited to see what would happen. and waited a grand total of an hour and decided that, cool, it must have been a dry bite, no effects. And off I went with a car full of venomous snakes. It didn't take me long to realize that I had, in fact, been envenomated and, in fact, quite severely, where the first symptoms were quite spectacular. One was my mouth basically turned to living metal. I got this intense metallic taste in my mouth. It's like all my tissues were made of metal, much like the Terminator and Terminator 2.
Hmm.
And then I lost my ability to see any color except for yellow. The world was white to black with an infinite degree of shades of yellow in between. And by now, I am absolutely delirious and totally lost somewhere in the farmlands of Oregon, a couple hours outside of Portland. Drove for I'm not sure quite how long until I finally encountered a little gas station convenience store in the middle of nowhere. Came in and said, "Could you please call an ambulance? I've been bitten by a venomous snake." Now, this is midwinter in the Pacific Northwest, one of the coldest, rainiest places out there. So it's about 38 degrees Fahrenheit and pouring down rain. So this is a highly unlikely statement. Instead, the gas station attendant just looked at me like I was another tweaker having a bad day
Yeah.
and sarcastically asked, "Are you sure it was venomous?" To which I responded non-verbally by having my head spin around 3 times and go the full exorcist while projectile vomiting all over everything. The
Oh.
counter, the lottery tickets, the cash register,
Oh,
before proceeding to collapse to the ground to have the first of 18 heart attacks that I would have over the next 90 minutes.
Wow.
oh,
So, yeah, that was a spectacular one.
yeah.
So we want to hear from you as the expert. We want to hear from you about the evolution of snakes, and there's a lot more to this story than just the fangs and the venom. But maybe before we get into those details, we know a lot about the evolution of snakes through the evolutionary record that we find in the fossil record, the skeletons in the rocks, but also genetic studies. So maybe just for a couple of minutes, just tell us about those two ways of getting information about the history of snakes and what do we know about them?
Well, the evolution of snakes is a classic example of where using morphology to reconstruct evolution is really a poor way of going about it. Snakes for the longest time were put external to lizards, but genetically, lizards are not a natural group. There are no lizards to the exclusion of snakes, just like there's no reptiles in general to the exclusion of birds. They don't form monophyletic groups. You've got snakes are actually nested deep inside the lizard clade, with the closest relatives being the iguania and anguimorpha lizards. And snakes themselves are just one of over 25 lineages of legless lizard. They're just the most highly tuned version. We have a bunch of other ones that are well on their way to becoming snake-like creatures. The major difference being that they lack the pelvic girdle. That's really about the only thing that defines them relative to any other group of legless lizard. There's a variety of legless lizards that lack ears. There's a variety of legless lizards that can't blink their eyes. Because we have legless lizards here in Australia that are evolved from geckos, which don't blink their eyes. So that like I said, there's nothing unique about snakes other than, like I said, maybe the pelvic girdle, some plasticity of the jaw, a bit more flexibility of the vertebra, but they're just points on a continuum relative to these other convergent evolutions. So it's, a lot of the ancestral views of snakes were driven by morphology, much like the idea that front fangs evolved in a stepwise manner, that you started off with rear fangs, then you went on to cobra-like fangs, and then you went on to the swinging fangs of a viper. But in fact, what we think of as hollow front fangs have evolved on 3 separate occasions within snakes. Cobra-style snakes evolved it independently of vipers, which evolved it independently of the stiletto snakes, the Atracaspis genus of Africa, where their fangs actually swing out on the horizontal plane. They come out sideways instead of on the vertical plane, like vipers. And that with all of those fangs evolved from the exact same teeth that become enlarged in so-called rear fang snakes, where the front fang snakes and the rear fangs, it's the exact same teeth. It's just in the front fangs, they've lost all the teeth in front. of those back teeth, and then you get a shortening of the bone and an elongation of another bone, pushing those teeth now more towards the front of the jaw. So a lot of, like I said, the historical views of it that were driven by morphology were completely wrong, where at the end of the day, DNA is everything, the rest is just details.
Right. And before you say anything about the DNA, the one advantage of those skeletons in the rocks is that you can actually date them. You can actually say how far back each one of these steps in the evolution goes.
Oh, absolutely. It's not to say that morphology doesn't have a tremendously important role, but when it comes to organismal relationships, it's a poor predictor,
Right.
just because you can't distinguish between what's convergent and what's divergent.
Right.
That is this a shared trait or is it something that evolved independently? Like the iguana lizards are the classic example where they were put down as a very basal lineages within the squamates, but when the genetics came out, they're actually very, very highly derived, and they're actually sitting right next to the monitor lizards and the snakes, which when we published that data back in 2006, caused, quite a bit of stir. The classic morphology driven taxonomy people were quite up in arms against this, but hey, the genes don't lie.
Right.
I'm the layperson in this conversation, and so I wonder if you could maybe simply explain how do genes help us be much more sure about relationships between species and so on than like you're saying morphology, for example. Like what is it about the genes that we can tell?
So with genes, think of it as sort of like Goldilocks. Some are too hot, some are too cold, and some are just right. But the just right one are actually a function of hot or cold based on the level that you want to look at. Different genes evolve at different rates. That some genes are very highly conserved and evolve very slowly. And these are typically ones that have a tremendously important function where any deviation from that function can lead to death. So
Okay.
things like hemoglobin or some of your cytochrome genes, your oxidase genes, these are ones that because they evolve very slowly are great for reconstructing deep time relationships. You don't want to look at something like an immunoglobulin gene because that's going to change very rapidly and you would become saturated. You'd have too many changes, it'd just be all noise. So if you want to look at deep time, like relationships between families or orders, really going deep into the organismal history, you want something that's slowly evolving. If you want to look at changes between species within a genus or even different populations within a species, like an island population that has been separated since sea level rise, that happened at a certain point, say 6 million years ago, that something like that would be appropriate to look at a more rapidly changing gene because then enough signal will appear. But if you use one of those slowly evolving genes to look at species, there's going to be little, if any, difference between them. So that's why picking which genes to use at different levels of time that you want to reconstruct is as much of an art as it is a science. And it has to be appropriate. And that's why there can be a lot of contention because it's not just the genes, but then how do you reconstruct it? How do you come up with meaning based on change? What is a change that might have happened independently versus a change that goes back to the base of that group? And there's a lot of different mathematical ways of reconstructing that, some of which are more appropriate for different levels of time or different degrees of change. And there can be a huge amount of debate and sometimes even leading to acrimony in between groups, at scientific conferences, where it becomes, in some cases, almost arguments of authority of which one is the most appropriate to use. And my simple philosophy when it comes to these sorts of things is looking for congruence, where if you analyze it using several different genes and analyze those using different mathematical models, if they come up with the same solution, then you know that you're on the right track. Versus if you have different genes that are supposed to be evolving at similar rates and they come out with wildly different answers, even within a mathematical model or between mathematical models, that should raise some flags, and you should be approaching things a bit conservatively at that point in time. So there's a lot of sort of steps that need to be cautiously approached before you can really make a lot of these sort of what may or may not be definitive statements.
And maybe we should talk about one of the first gene changes, one of the first genetic changes. That's what we're talking about now. I mean, the obvious thing that we need to say about snakes is they don't have legs, and I think we believe they came from four-legged predecessors, they're lizards of some kind. So maybe just say a little bit about how they lost their legs and why would they? I mean, that seems a little bit radical and maybe even short-sighted to just drop off your legs. But there are some ideas as to why they would do this.
Yeah, one of the earliest ideas was the aquatic ancestor idea, where snakes lost their legs because of locomotion in the water. Now, a problem with this is that there's not a single lineage of legless lizard out there that is aquatic. And if we look at aquatic animals like mosasaurs that evolved from monitor lizard-like animals, they retain their legs. So that was a big sort of X against that theory, even though you do see it kicking around. But rather, what we do see when we map the morphology and habitat of all the different legless lizards out there that we have today, all the non-snake legless lizards, is that we see them bunching up into two different groups. One is these long-tailed grass dwellers, and then these short-tailed burrowers, where if you're swimming through grass, having a long tail is something that's very good for propelling you. Well, if you're burrowing underground, a long, thin tail is not going to allow you to get the force to push through the substrate. So one is swimming and one is pushing. And we see all of our legless lizards falling into these absolute groups that way, that it's always that way. So with the case of snakes, since they're a very long-tailed animal, if we go back in time and look at our basic snake morphology, that it would seem that the earlier snakes were likely grass swimmers, rather than burrowers. They're sort of semi-fossorial or moving through leaf litter and grassland, using this long tail as a form of rapid propulsion. And it's fascinating if you look at the genetic difference between our long-tailed grass swimmers and our short-tailed burrowers, that the long-tailed grass swimmers unsurprisingly have a much wider genetic range typically than our short-tailed. Like our short-tailed ones are pushing through a substrate, so they're under a strong mechanical selection pressure to where their head shape is going to be dictated by the type of substrate that they're moving through. You're going to have a different head shape if you're moving through fine sand than clay, than sort of more rubbly type. As far as your sand one, it's going to be a long, pointy head, like we see with some of our sand swimming lizards, versus our ones moving through more robust substrate are going to have more almost like a bulldozer type head. It's going to be blunter and very heavily reinforced with bone in order to get through that substrate. So we see much more genetic diversity in our burrowers because they're going to be very highly adapted to that particular habitat. Like, for example, in California alone, there's 6 different species of burrowing legless lizard there, each which is specialized for a different type of substrate.
Now, the loss of limbs in snakes isn't complete, or at least not in all snakes. Some still have little bits of stumps, little bits of pieces of, hip bones and whatever, I seem to understand.
Yeah, you see this in some of the basal snakes, like in some of the pythons, and the males are actually able to still wiggle those remnant back legs. And there's a sort of a theory that they are using it to kind of like tickle the female during the breeding process, because you could see him moving it back and forth. He could also just be moving it back and forth because he's really happy. But, so whether or not it's actually used that way, hasn't quite been proven. It's, an amusing idea that it has. But yeah, so they still have residual back legs, these relic structures that there's still muscles attached, and they can, like I said, move it, these little conical stumps back and forth.
Yeah, we see the exact same thing in whales and dolphins. They've lost their legs, and you still see stumps, hip bones, or little pieces of hip bone in them.
Cool.
And when I was thinking about that, it occurred to me, T-Rex is a lizard, is a reptile, and it's got these tiny little front arms. And the question just popped in my head: was T-Rex on its way to becoming a snake?
Well, I don't know about a snake, but certainly the losing of the front legs makes perfect sense from a biomechanical perspective, because if you've got a big head and you're on a body where it's on a horizontal plane, where the head is on the same plane as the tail, you need to have a tail to counterbalance the weight of the head, but it's still under strong selection pressure to minimize the weight on the front end as much as possible. Losing limbs is an excellent strategy for that, which is why we see that over and over with our big carnivorous dinosaurs, that the larger they are, the more likely they are to have dramatically or almost completely lost front limbs. But like if you look at the T-Rex babies, they start off with the front arms proportionally much longer. They almost look sort of normal. They almost look more like a raptor early on. But as they age, the front legs or arms, whatever you want to call them, don't grow to anywhere near the same degree as everything else in the body. So eventually they end up with these undersized little arms. And like with T-Rex, it's a great debate of, are they even using it anymore? The bones suggest that they were using it to some form. They're to grip on, help push themselves off the ground, whatever. But the bones are short but quite robust. There's suggestions from the bone morphology that there was very strong muscles attached to them. But in other lineages, like some of the carnosaurs, the front legs are pretty much, or front arms, like I said, whatever you want to call them, they're almost completely gone. So very different evolutionary trajectory. But it just comes down to the counterbalancing. Same sort of thing we see like with kangaroos, where they've got dramatically reduced front arms, because of the need for the counterbalancing.
Okay. I did a little bit of background on this, and I found there's some ideas that people have as to how this happened, how they actually lost their legs. It was one particular study where it's a genetic switch that they found in these animals, which regulates the development of the legs of these animals in the fetal stage. And they took this genetic switch, they swapped it in and out of mice. They swapped in the same switch that you might find in humans or in fish, I think it was. And these mice have normal leg development, but they swapped in the same switch, a piece of a DNA from snakes, and these little mice suddenly had really, really shriveled up limbs. And they looked a lot more streamlined, like snakes.
Well, what's interesting is that with those genetic switches, that the front and back legs are under different control. Like we have some lineages of legless lizards that have lost the front legs and others that have lost the back legs.
Okay.
So it's fascinating that it's not an all or nothing between the two limbs.
Right. And so I'm wondering if this story that I was reading about, if we're talking about the same thing, or maybe this is a gene that's even upstream of the ones that control front or back, that maybe this is a gene that controls both, I don't know. I just know that they swapped in these sequences from humans, fish, mice, and snake, and they found that when they put the snake gene into the mice, they basically, they had barely a stump. And for the nerds out there, the gene that they were talking about, they refer to it as sonic hedgehog. I don't know if you want to tell a bit about.
Well, there's a lot of sonic hedgehog genes out there. It's one of the more popular developmental biology acronyms. Like with the snake fangs, a study that we did that was published in Nature in 2009 showed that the snake fangs are controlled by a sonic hedgehog gene, and it's the exact same gene that does the development of the enlarged teeth in rear fang snakes, and it's the same gene that's turned on for the fangs of a cobra or the fangs of a viper.
Now, I don't want to talk about the teeth just yet, but I also read that these sonic hedgehog genes, they're involved with the number of vertebrae and the ribs, whether each vertebra has a rib or not, and that too you see in snakes. Snakes have a lot more vertebrae. They have a lot more ribs, and that's also under control of the same set of genes. You want to tell us a bit about that?
Well, like I say, the plasticity that's out there is extraordinary. Like with snakes, that you can tell a snake from a legless lizard just by holding them, because as you rightfully pointed out, they have a lot more vertebrae, so they've got a lot more flexibility. So even the most snake-like, non-snake legless lizards out there, like some of the ones here in Australia that from a meter away, they look just like a snake, extraordinarily long tails, quite flexible for a legless lizard. But when you pick them up, they still are a little bit stiffer than a snake because they've got 30% fewer vertebrae than a snake. And of course, having more vertebrae is one of the things that gives the snakes their agility, such as the ability to do extraordinary climbs. Like the way that like a snake goes up a tree by wrapping around the tree and then sort of pulling itself up, along the way. That flexibility and the range of motions that they have is just remarkable from just, an athletic perspective. and having more vertebra gives them that suppleness. So yeah, it's fascinating to think of something like hard tissue, like bone and the number of vertebra, being something that's very highly plastic and that can be shaped by these different evolutionary processes in short periods of time, where turning something like that on and having suddenly 30% more vertebra would have been a classic key evolutionary innovation, which is that idea of a mutation happening that is just like a quantum leap forward. Has to do with that idea put forward by Stephen Jay Gould of punctuated evolution, where you just get something happening and then just boom, you get this rapid radiation as a consequence. So early on in snakes, when that mutation happened into a sonic hedgehog, and sometimes it could be just a single point mutation in a single gene, can trigger a radical morphological
Right.
change, and then that would underpin the explosive radiation of that lineage.
Right.
It's interesting. I do some 3D printing, and one of the most fun things to print are snakes, and kids really like them, and they really demonstrate that, like that articulation and just all the different vertebrae. If you print a long one, it's just amazing how you can just hold it in your hand and it'll just slide around. It really shows that.
Okay. Now, let's get to the fun part, the teeth. Snakes have very unique teeth, and some of them, it's very exaggerated, very long fangs pointing backwards, and they're hollow. That's a whole evolutionary story in itself. So maybe tell us a bit about that.
So it's the teeth came later in the evolution of the venom system. They were the final refinement because, of course, you wouldn't evolve an intricate structure like a hollow front fang in the absence of something already being there that's worth developing. So that's why venom came first in that primitive state where the venom glands are highly modified saliva glands that are very promiscuous, where they're a very leaky form of tissue, where if you look at like the transcriptome of any venom gland, way deep down in your under 1% level of expression, you're going to find all kinds of proteins, none of that are normally expressed elsewhere in the body. And that's where all of our venom gland components have come from. So the earliest venom components, like CRISP and kallikrein type serine proteases, were types of proteins already present in the saliva that became weaponized. But then subsequent forms of toxins were from this leaky expression. So we have ones that are normally expressed in the pancreas. You have other ones that are normally expressed in the liver or the heart. Or in the case of some of the Australian elapids, where they get like the taipans and brown snakes and tiger snakes, they get their devastating potency because they're actually secreting active forms of blood clotting enzymes. So it's like factor 10A, for example, where the pathology is no different than if I took a whole bunch of human factor 10A and injected a person with it, they would have the exact same pathological outcome as being bitten by a brown snake. So it's this overdose scenario. But it's a good example of that leaky expression where, as with anything else in evolution, if it confers an advantage, however minuscule, that trait becomes fixed and then amplified. So in the case of factor 10A, the clotting enzyme being recruited for use as a toxin, that occurred at the base of the Australian snake radiation subsequent to the invasion from Asia, where the elapid snakes encounter this virtually snake-free continent. So boom, extremely rapid radiation across the continent. But before that happened, virtually the very base of it, 10A was recruited as a toxin, but it wasn't amplified immediately. It was amplified on 7 independent occasions within the Australian lineage, within different genre. And say, like in some cases, like the black snakes, a single species has amplified it. And that trait gives all of them this powerful procoagulant mechanism. So getting back to it, the way to think of snake venom evolving, or any venom for that matter, is that you have these promiscuous glands that just randomly are expressing genes normally expressed in other parts of the body, something that doesn't happen in other tissues. Like you don't want your pancreas to start popping out something normally made in your brain or made in your liver. This, of course, is what happens with cancerous tissues. Why cancer kills people is that cancers are excreting proteins from all over the body. So you have pancreatic cancer, it might be pumping out neurotransmitters from your brain. And of course, no good is going to come from that.
Yeah.
But in the case of the venom glands, there's no downside So it's completely removed from that normal regulatory control. And then if some random event like the neurotoxicity of cobras and other elapid snakes is actually from the secretion of a brain neurotransmitter that's now been weaponized and increased in its potency. Same thing like with the factor 10A that we see in the Australian snakes, that their action on prothrombin, converting it to thrombin, is over 1,000 times the rate of normal factor 10A. So you get this initial excretion of it, and then it becomes amplified, and then under selection pressure to become even more potent. And same thing with these brain neurotransmitters that are now in death adder, cobra, right, mamba venoms, that they bind to the postsynaptic nicotinic acetylcholine receptor with a dramatically higher affinity than the normal brain neurotransmitter version.
So if I could just summarize that for the listeners, because there was a lot of information in there. You said that these predecessors had salivary glands, as all higher animals have, but you referred to this leaky expression in the salivary glands. Expression meaning they make specific proteins, and leaky meaning they make mistakes in which proteins to manufacture. So in addition to salivary enzymes, these animals were also making small amounts of other proteins that are normally made in other parts of the body. But that didn't matter because those mistakes were just ending up in the saliva and eventually the stomach where they got digested. But if some of those mistakes had useful effects, like, a brain toxin or a hyper blood clotting protein, and you can introduce that into the bloodstream of the prey by biting it, well, that becomes really useful, evolutionarily useful. And so these mistakes became permanent, the expression got amplified, and those salivary glands transformed into venom glands.
Yeah, so you can think of it as that the venoms are going to cause one of two effects. One will be an overdose scenario, such as the factor 10A clotting enzyme in the Australian elapid snakes, where it's like we've overdosed on our blood clotting, and we're now completely outside of regulatory control, and hello, massive blood clots forming in the body, which would rapidly immobilize a prey animal through stroke. And as we all know about stroke, it's an incredibly incapacitating event where a prey animal can't run away or fight back if half of its body is paralyzed by stroke. And it's obviously going to succumb quickly after that. But venom is not about killing. Venom is about prey immobilization. So immobilizing by stroke. So there's no functional difference between an animal that's fully conscious, unable to move, or one that's unconscious, or one that's dead. They're all equally swallowable, and they can just suffocate in the stomach. The other main action of venom is to cut lines of communication so that things shut down. And that's where like the alpha neurotoxins of these modified brain neurotransmitters that we have in cobras, kites, mambas, death adders, those sorts of snakes, they bind to the postsynaptic nicotinic acetylcholine receptor, which is the receptor on our muscles that receives the neurotransmitter acetylcholine telling it to move. So you wouldn't know that toxin is even sitting on that receptor unless you try to do something like move or breathe. And then you're very quickly going to know that it's there because you can't do either of those. So it doesn't stimulate an action. It just blocks that chemical email from getting through. And if you can't move your diaphragm, you're not going to have any oxygen exchange, and you're toast.
Right.
And similarly, if you can't move your legs, you can't run away or fight back, and you're toast.
Sorry, just curious. Again, the layman's, just listening here. The development of the venom doesn't obviously affect the snake. Why is that? It just sits in the gland. It doesn't enter the bloodstream, or?
Well, two answers to that. First is yes, the gland, anything that's inside the gland is actually outside the body. If you look at the cavity of the gland that empties into the mouth, that cavity is not in the body anymore.
Okay.
But snakes in general are, of course, still immune to their own venom because otherwise they do something like bite their lip, no good is going to come from that. That's one of the things we look at in my lab is the evolution of venom resistance, both within the venomous animals and also the prey of those animals or predators of those animals. So we've shown that there's a number of different mechanisms for venom resistance, and that some of them have evolved convergently across both predators and prey of venomous snakes and the snakes themselves. Like we showed that the slow-moving terrestrial pythons of Africa and Asia are resistant to cobra venom through the exact same mechanism that a honey badger has its iconic resistance to cobra venom. That's through this electrostatic charge repulsion, where the target of the alpha neurotoxins, the postsynaptic nicotinic acetylcholine receptor, in its natural state is a receptor that has a strong negative charge to it. If you look at the amino acids that coat the inside of the receptor, there's some invariant ones that are like glutamic and aspartic acids that have a negative charge to the receptor. And that's something that helps guide our normal binding of acetylcholine. So snake alpha neurotoxins have evolved to have a strong positive charge. So the initial binding is guided by this opposite charge attraction. So with the evolution of resistance to that, some of these pythons and the honey badger have flipped the charge state of their receptors, where their receptors now have a net positive charge. So it's like trying to bring the same side of two magnets together. You can't. There's this electrostatic repulsion of the neurotoxins. They just cannot bind to the receptors. Another major way of being resistant to snake venom neurotoxins is to have what's called N-glycosylation, which are these long branched sugar groups emerging from the inside of the receptor. That think of them as like shrubbery, where the small acetylcholine molecule can still get past that, but the much larger neurotoxins are sterically hindered. They're physically blocked from being able to bind to the receptor. And we see this evolving independently. This is how cobras are resistant to their own neurotoxins. But we also see it, like, for example, in the mongoose with a cobra predator and how it's resistant to cobra venom. So both the animal itself and its predator have evolved the exact same mechanisms of resistance. And then these kinds of evolutions of resistance in predators or prey could then trigger selection pressure for new types of venom. So, for example, the long-glanded blue coral snake of Southeast Asia, Caliopsis bivirgata, look it up. They're stunning. They're these electric blue snakes with a bright red head and a bright red tail, and then black stripes running down the body. They look like a cartoon, but they're a predator of predators. They feed on other types of elapid snakes. So using the normal type of elapid venom wouldn't allow them to do this, but there's a mutation that happened in the common ancestor of this genus, allowing the venom to bind to sodium channels instead, on the presynaptic side, and triggering a spastic, rigid paralysis instead of the normal limp one. But because it's got a new target, that means that any elapid snake out there is fair game for prey. So they're eating cobras, they're eating krites, they're eating little king cobras. So, it's a predator's predator because it's got this novel venom phenotype.
Wow.
So again, if I can summarize that, because I'm loving what I'm hearing here, but I'm understanding a lot of what you're saying. So I'm going to try to summarize really quickly what you said there. 3 basic parts in your answer to Scott. Why doesn't the snake die from its own venom? And you said, first of all, the venom in the saliva, when it's in the saliva, it's not dangerous. I mean, indigenous peoples can paint their arrows with the venom of these animals, and they can eat the meat that they catch because the venom is in the saliva. It's in their gut. The venom isn't dangerous when it's in the saliva or your gut. It's only when it gets into your blood that it becomes a problem. And so that was the first reason that you gave. The second reason is that the snake makes actual physical changes to the targets of the venom in a way that the venom can no longer find a target. So I think that was the 2 reasons you gave. I'm wondering, though, Bryan, isn't there a third one? Don't the snakes develop an immune response against the components of the venom?
Well, with the resistance, it can be immunological, like we've shown with some of the Australian lizards that are resistant to blood-acting toxins. They have proteins in their blood that bind to the venom and neutralize it so that venom can't act on the blood, like red-bellied black snakes trying to feed on blue-tongued skinks. The blue-tongued skinks are able to mop up those toxins, and it doesn't affect their blood. Or it can be physical changes to the target itself, like modifications of the nerve receptor itself in a myriad of different ways. And we've shown this happen so many times in different lineages, like with the Australian skinks, which were under enormous selection pressure from the elapid snakes here. evolution to venom has evolved on 25 separate occasions just within the Australian skinks, or the caecilians, the legless amphibians that would just be fettuccine for the snakes to slurp down, we showed in time and space that as elapid snakes migrated across Asia and into Africa and then across the Bering Strait and down into South America, that as they evolved, it would have been Armageddon initially for the caecilians, but there was a strong selection pressure for the evolution of resistance. And we showed that with caecilians that they've evolved venom resistance on over 20 separate occasions. And sometimes lack of resistance can be as informative as evolution of resistance. So in the case of Africa, where all over Africa, there were a myriad of different independent evolutions of resistance because of cobras being all over Africa. But then the caecilians that we sampled from the Seychelles Islands, which are elapid snake free, there was no evolution of resistance within those caecilians, showing that it is due to the selection pressure of the snakes that they've got these modified receptors. But then that puts a pressure back on the snakes. Like in South America with the coral snakes, they have extraordinary diversification of their venoms in response to the evolution of this resistance by their prey, where now the venom's going to attack all different kinds of targets besides the one that they can't hit anymore. So it's this constant battle between predator and prey in this chemical arms race that's the classic example of the Red Queen hypothesis of running, never getting away, or running, and never catching up. That it's this dynamic change, that any change in the venom puts a pressure on the prey, and then correspondingly any evolution of resistance in the prey puts a pressure on the venom. So it's this fascinating interplay, but all of that leads to the great big pile of it sucks to be us, when it comes to the clinical effects, because that dynamic diversification is the silver lining that gives venom that great potential for drug design development because of this almost infinite degree of novel bioactive compounds. But the dark cloud inside of that silver lining is that you can have radical changes in the venom, so much so that even between closely related populations of the same animal, you can have the antivenom work on one, but not the other. The one is that, like the Southern Pacific rattlesnake that we're working with outside of Los Angeles, that on the desert floor, there are blood-acting toxins in these snakes, and Crotalus helleri, that it's like basically Ebola with fangs. But then when you drive up into the San Jacinto Mountains, it switches from an anticoagulant venom to a neurotoxic venom that the antivenom doesn't work very well against. And then as you keep driving over this is on the wet side of the mountains, you get this neurotoxic venom. But then when you keep drawing now into the inland, the dry side of the mountains, into the high desert, that it now switches back to a coagulant acting venom, but instead of being anticoagulant, it's now procoagulant. It's turning on the blood clotting. And again, the antivenom doesn't work. So this is all within a 6-hour drive. You have 3 different types of venom from virtually genetically indistinguishable snakes, because remember that Goldilocks thing that, venom evolves even faster than our immune proteins. Like that classic idea, dividing number of changes that make a change versus the ones that don't confer a difference in amino acids. So you're non-synonymous by synonymous. If you end up with more mutations that make a change in amino acid than less, you end up with a omega value of over 1. Under the neutral hypothesis, everything should be 1. Something that's under negative selection should be less than 1. So like our immunological proteins are cooking with gas. You get omega values of 2 or 3. And we consider that the benchmark for proteins that are evolving under accelerated evolution. Venom proteins can have omega values of 4 or 5, or even up to 6. So extraordinarily highly evolving proteins. So that means that while the snakes themselves aren't different species, the venom can be radically different, like I said, just within a 6-hour drive. Wow. And this has a huge public health problem because antivenoms are typically made using just a very restricted range of the snakes, often whatever is the easiest to collect, but that isn't necessarily what's biting people. And then it gets even more complicated where baby snakes can feed on very different prey than adult snakes. So just within the lifetime of a single snake, its venom can change radically. Like, for example, the Australian brown snakes, babies are nocturnal feeders on sleeping lizards. So they've got a neurotoxic venom. As they age, they become daytime active hunters of mammals, and they've now got a procoagulant venom. All the antivenom is made using the venom from adult snakes, which means it has zero action on the baby venoms. Completely different venom phenotype. So if you're bitten by a baby brown snake, it's probably better to actually get death adder antivenom, which is made against snakes with highly neurotoxic venom. You'd have a better chance of it neutralizing than you would from brown snake antivenom made using the venom of the adults. So that's just the most extreme example, but we see this time and time again. Like one of the snakes we're working with, one of the European vipers, the babies are procoagulant while the adults are anticoagulant, and they're acting on the exact same target, just different forms of it. The babies are turning factor 10 on, leaving the zymogen 10 into the active form 10A, while the adult venoms are actually blocking 10A, the active form within the body. So one of them will cause a stroke and the other one will cause uncontrollable hemorrhage. And obviously the antivenom is only going to work against the one that the antivenom is made using, and that's the adult snake.
So what I'm hearing is the hope for some sort of universal antivenom or at least maybe dozen antivenoms that apply to many cases. That's not going to happen.
No, it's not. But one thing that is showing great promise is the repurposing of enzyme inhibiting drugs, like phenomastat and marimastat, for example, that block metalloproteases. And that's one of the major toxin types within snakes. While there's been over 30 different types of proteins that have been weaponized for use as toxins, venoms are still dominated by 4 or 5 major ones. And sometimes you may have just one of these making up the entire constituent of the venom. Now, you can have a lot of different forms of metalloprotease in there, but they're all still metalloproteases. They'd all still have that core element that defines them as an enzyme class. So that becomes the kryptonite angle, where the enzyme inhibiting drugs can become almost universal against any kind of metalloprotease, whether it's one that's turning on a blood clotting enzyme, one of them that's destroying a blood clotting enzyme, one of them that's turning your arm into black goo, because it's a voracious necrotic enzyme. All of those are still metalloproteases, and all of those still have the same core vulnerability. So we're doing a lot of work in that angle in our lab, as are a number of other research groups worldwide. And we think that this may be the smartest way of going about it, is to attack the enzyme class rather than attacking the function.
In the case of the brown snake, I'm just curious, but would they ever give both? If somebody's bit, they don't know which one, the adult form or the younger form, would they give both antivenoms?
Well, this is why in Australia, I'm an advocate to actually ditch the use of monovalents, which are antivenoms made against a single type of snake. And we have a very good polyvalent here. It requires a higher protein load, which I think is not as big of a problem as it's made out to be. But the advantage of the polyvalent is that polyvalent's got death adder immunoglobulins in it. It's got immunoglobulins, there's going after tiger snakes. It's got ones after taipans, sea snakes, brown snakes. So it's got every venom phenotype is rolled up into that one larger vial. So it gives you a more comprehensive neutralization capacity. The whole idea of monovalence is to restrict the amount of protein and therefore trigger immune reactions to foreign proteins. But the antivenoms nowadays are so purified and so clean, this is less of an issue than it was 30, 40 years ago, where the foreign protein load really triggered a lot of allergic reactions. But a lot of that was actually due to non-immunoglobulin proteins. It was due to the antivenoms being poorly fractionated, where there's a lot of albumin, for example, in there. That's what that was actually triggering a lot of the allergic reaction, was not the immunoglobulins themselves, but the non-immunoglobulin proteins. And then also nowadays we have the antivenoms, like in Australia, that are processed to remove the part of the antivenom that screams force. So if you think of how an antibody is shaped, view it as a Y. Now the stem part of that Y down at the bottom, the FC region, doesn't participate in the interaction of any sort when it comes to immunology. Rather, that's the part that identifies it. Did it come from a rat, a horse, a human, whatever? So in Australia, the antivenoms are cleaved, so that part's removed anyway. And then they're very highly purified. So there's basically little or no non-IgG protein in there. It's almost completely immunoglobulin material. So that's already reduced the foreign protein level and reduced the tag that says horse. So I think giving a larger vial is not a downside because I don't think it's going to trigger much of a reaction anyway. But the upside is that you have a more complex range of immunoglobulins in there, and therefore a greater chance of picking up something that's weird or an age-related change like the neurotoxins of the baby brown snakes.
So we're over an hour now, and I had a couple more questions, but I just don't want to take up too much time. So I'm going to at least put them on the table, and we'll see if we can do them very quickly. The first one would be, I wanted to hear something from you about how we talked about the development of these venom glands and then the venoms that are in them. We talked a bit about the teeth, but the one part of the teeth that we didn't talk about is how these teeth became hollow and shaped like syringe needles. Can we do that in a minute or two?
Yeah, so there's rear fangs, the enlarged rear teeth have happened on quite a large number of occasions. So first off, that creates the wound that's necessary for venom to enter because venom proteins are too big to enter directly through the skin. And then if you have channeling, if you have grooving of that teeth, that helps direct the venom a bit further. And then sometimes you get really deep grooving, and then that helps it more and more. And eventually that grooving will start sort of sealing. And we have rear fang snakes that are so deeply grooved that it's almost sort of like a proto-hollow fang. So you can kind of see that you grooving and then forming a tube, that tube could still be open at the top, but then a stepwise evolution to sealing that tube. And then, and then you would then have muscles to propel the venom down that tube. So the muscles that propel the venom have evolved on more times than hollow fangs, as you would expect. So we have these compressing muscles that compress the gland associated with all of our hollow fangs. But with some of our rear fangs, like the African boomslang, has muscles that contract its gland, and they've got deeply grooved teeth. So we can see these sort of pumps already happening. So grooving and then a pump, and then once you have a pump, there's now a selection pressure to increase the grooving, and then eventually, there's a more and more of a selection pressure for that grooving to eventually become sealed. So it'd be you can view it as a stepwise of enlargement, grooving, pumping, sealing into that hypodermic aspect.
Okay. You started that answer off talking about, rather than the hollow fangs, you began talking about just a groove, a channel, and the snake begins to channel the venom in the right direction. So the second question was this. I know that one of the most recent innovations in snakes is these spitting snakes. They don't bite and inject. They actually spit their venom up to 10 feet or 2.5 meters. And there's a bit of a story about that, an evolutionary adaptation story to that too.
So first of all, I hate the name spitting. It should be squirting because it's a squirt like a syringe.
Right.
Anyway, so with the spitting cobras, it's this fascinating complex interplay where they've got these pain-inducing toxins of different kinds. And we showed that the ability to destroy tissue and cause pain that way was linked to the hooding, where the hooding came first as a form of warning, but then when you start getting more intricate markings on the hooding, that's shown to be linked to an increase in the pain-inducing defensive toxins. And this happened before spitting. So the pain-inducing toxins evolved before the spitting cobra, and then the spitting became a further refinement of that, but that evolved on 3 separate occasions within the cobras, and that's been linked to the spread of the hominids. But what's fascinating is that, with the spread of the hominids putting a selection pressure on the snakes have also put a reciprocal selection pressure on the hominids. So across Africa, the African primates are more resistant to cobra-style venoms than any other lineage of primate out there, much more so than the Madagascar primates, which are in a cobra-free environment, or South American primates, where the elapid snakes there are nocturnal and fossorial. So they have a big interaction with these daytime and active snakes. And then with the evolution of bipedalism in the last common ancestor of chimps, gorillas, and hominid lineages, that there was an increase in the venom resistance. So chimps, gorillas, and humans are actually much more resistant to cobra venom than other lineages of African primates. So those African primates, in turn, more resistant to snake venom neurotoxins than other lineages of primates. So it's this fascinating interaction between the snakes and the primates, and that with the evolution of spitting, there was a further independent amplification within each of those lineages for the pain-inducing toxins. So we've got pain-inducing toxins evolving early on with the evolution of hooding, further amplification of them with the snakes that become brightly colored with warning coloring, your classic aposematic warning coloring, which has evolved on a myriad of occasions in Africa and Asia. Some of them, for example, like they look like bumblebees, and that's evolved on 3 separate occasions. Others, the use of bright red coloring, that's evolved on multiple occasions as well. And then you have the independent evolution of spitting with an amplification of these pain-acting toxins occurring within each of these. But then again, like with that fascinating interaction with the primates themselves of becoming resistant to cobra venom and then that becoming amplified further within the hominid and apes and chimps last common ancestor.
So I was really fascinated with that part of the story, that these snakes went through hundreds of millions of years evolving to just bite animals and inject their venom. But now there was this recent change. They encountered a whole new species, the hominids. And the first thing the hominids did when they saw snakes was they'd grab a stick and start beating the snakes. So the snakes very rapidly developed this behavioral evolutionary response where they learned, rather than trying to get up to the hominid and bite them, I'm going to spit them from 10 feet away. That's that whole evolution of a behavioral response. Unfortunately, it was at this point in our interview that Bryan was suddenly interrupted by something very important that required his immediate attention, but he was polite enough to give us 2 more minutes to wrap the interview up. So, Bryan, we really appreciate this. This has been an incredible story. So many other things we wanted to talk about, but just this whole idea of the evolutionary journey of the snakes, so complicated. Their legs, their vertebrae, their ribs, their teeth, the venom glands, the ducts, and even the venom itself. It's not a simple thing to become a snake. It's a fascinating story.
Thanks.
That'd be a good headline. It's not a simple thing to become a snake.
Yeah.
Well, thank you so much, Bryan. We really appreciated this.
No problems at all. Send me the link when it's up and I'll put it up.
Okay.
Thank you.
All the
right,
best.
have a good one.
Thank you.
All right, take care.
You've been listening to Recovering Evangelicals with your hosts, Luke Jansen and Scott Dyer. Send your comments and suggestions for new episodes to lukejjansen@gmail.com. Join our private discussion group on Facebook by searching for the Recovering Evangelicals podcast group. Look for the logo that you always see at your favorite podcast catcher and ask to be let in. Subscribe to lukejjansen.wordpress.com if you want to get advanced releases and group posts, or browse through our archive for a full listing of all our previous episodes. And please help the podcast by sharing with a friend and leaving a review at your favorite podcast catcher.